What is a litmus paper? Litmus paper is used in laboratories to detect the presence of acids and bases, alkalis. Acids turn blue litmus red, while bases turn red litmus blue. Neutral solutions, neither base nor acid, do not change its color. A common laboratory practice is to dip pieces of litmus paper into solutions to detect if they are acidic, basic or neutral in nature. Do you know what this litmus paper is? Litmus is a natural dye made from small plants called lichens. These plants are of several varieties and grow in abundance in the Netherlands. When a lichen called Richella tincotoria is allowed to react with ammonia, potassium carbonate and lime, it gives a blue color material which is used as the coloring matter for paper. The paper is dipped into it and dried. This is what is known as blue litmus paper and used to test acids. Orchil or cudbear is a red dye obtained from another species of lichens. This is used to make red litmus paper. Alkanet or alkana is another dye obtained from the root of the plant Alkana tinctoria. The coloring ingredient, alkanin, is soluble in alcohol, benzene and ether. When white paper is impregnated with an alcohol solution of alkanet, it becomes red. This paper is turned to blue shade by alkalis. When a chemist wishes to neutralize an acid solution, he first adds litmus. This changes its color to red. Then the base is added until its color changes to violet. The solution then becomes neutral. When acids and bases react, they produce salts by neutralization. We make use of various salts in our daily lives. The common salt, used as a food material, is a kind of salt which can be produced by the reaction of caustic soda and hydrochloric acid. Nowadays, litmus paper is made from several substances such as azolitmin, crystrolitmin, spanulitmin, etc. These are apparently mixtures of closely related compounds that were identified in 1961 as derivatives of the heterocyclic compound phenoxazine.